So the Physiology of Obesity Mechanisms to Medicine series started a few years ago when myself and co-organizer Dr. Joe Lewis at the University of Cambridge felt there was scope to develop an ECR-focused symposia, which could give more speaking opportunities to ECRs and bring those of us in the obesity and metabolic disease field together to network and disseminate results. After 3 successful symposia at the European Congress of Obesity, Experimental Biology, and a day at Physiology 19, we're delighted to now bring this into a webinar series to give fellow ECRs the opportunity to present and network during a time when conferences are being cancelled and opportunities to do so are minimal. With over 1,400 registrations across over 14 countries and another 1,300 views on YouTube in the first 6 sessions, it's great to see so many of you here as we begin the adipose tissue and metabolic physiology session. So for this half of the series, Joe and I will chair alternate sessions with an invited ECR co-chair each week. Today, I'd like to welcome co-chair Arianna Fazzato, PhD student in Dr. Beatrice Filippi's lab at the University of Leeds, where her research focuses on mitochondrial dynamics in the dorsovagal complex, feeding behavior, and energy expenditure. And now over to Arianna with some housekeeping, and who'll introduce the speakers.
Hello, everyone. So before introducing the speaker, I'll go through some basic housekeeping rules. So our speaker will be presenting their work for about 20 minutes, and then that will be followed by a 15-minute question and answer session. Please submit any question through the Q&A button at the bottom of your screen, and the question will be directed to the speakers during the Q&A portion of the webinar. And please do not submit questions at any time, as we will be looking at this through the presentation. You have the option to submit questions anonymously if you like to do so. If not, please include your institution at the beginning of the question, so I can announce this with your name. And we might not have time for all the questions, but we will try to go through as many as possible. You can upvote the questions as well, and you could type your own answers or comments to other attendees' questions. And please note that this webinar is being recorded and will be available for rewatch. So I will now present the first speaker for today's seminar. So we're going to hear talking Dr. Bruna Brandao from Dr. Ken's lab at Joslin Diabetes Center at Harvard Medical School in Boston, who will present her work on adipose tissue, microRNA, and exercise performance. So I'll mute now and let Bruna speak.
So first of all, I would really like to thank all of you guys for the invitation. I was really surprised and happy to be here and be able to share a little bit of the work that I've been developing for the couple of years. It's a done work, and I'll just go over some data and hope that you get excited as I was when I was doing this work. So we're going to talk about adipose tissue and microRNA and how it can modulate exercise performance. First, but before going to the data, I want to acknowledge many people that helped me to develop this work. Dr. Marcelo Mori, which was my mentor during my PhD. This work was mainly done during my PhD time. And all the members, actual members and former members that helped me with all the experimentation. Also, this is a shared work with Jonas Tribeck from the University of Copenhagen. So Soren, it's my co-first author, which helped me a lot. Well, we worked together to develop this work, and all these people that are in here that I could not have this done without the help of everyone that is in here. And mainly also Dr. Khan, because he allowed me to perform some experiments, even though I had moved from Brazil to Boston. So I was I'm at his lab now, and he also helped me to develop some experiments in here. So I really like to acknowledge all of these people, and very thankful. And of course, all the financial support that were involved in this work. So I just want to give a brief overview of my career timeline. So I have background in physical education or sports. I have a master's degree in physiotherapy. And in 2012, I started my PhD in molecular biology at Federal University of São Paulo from 2012 to 2016. And at that time, I also had the chance to go as a visiting student to the University of Copenhagen, where I was mentored first by Sarah Wingberg, which is working now at Novo Nordisk, and after that, Jonas Trebeck. When I finished my PhD, I moved to Boston since 2017. I am a postdoc fellow at Dr. Khan's lab at Joslin Diabetes Center. All right, so here I will just give a brief overview of how acute or chronic exercise can influence adipose tissue metabolism. So we can share this effect in two, as I said, acute, where we're gonna see the triglycerides breakdown and race reification. This mechanism is quite interesting because as you increase lipolysis, you also increase race reification in a way to protect the adipose tissue to lose all the triglycerides that is on it. We also see the secretion of signaling molecules. We now know that many proteins and lipids can be secreted from adipose tissue, and this has a role in exercise metabolism and physiology. And we can also have chronic effects, which comes after many sections of exercise, which is characterized by decrease of adipose tissue adipocyte size, and also increase of adipose tissue plasticity. And to regulate all these different metabolic pathways, the cell needs to have a very refined mechanism of gene expression and protein abundance control. And one of these mechanisms, it's the microRNA pathway. So microRNA are considered as fine-tuning mechanism to regulate the cellular proteome. So basically, here's the microRNA processing pathway, which starts in the nuclei where the pre-microRNA is transcribed. So we have this hairpin double-strand RNA bound by this tail of nucleotides. This combined hairpin double-strand RNA will then be processed by DROSH and DGCR8 and will become a precursor microRNA, which will be exported to the cytosol. And in the cytosol, Dicer will come and remove this stem loop in here. And now we have two mature microRNA strands. Very often, one of them will be degraded and will not regulate gene expression, but we've seen example of microRNAs that either the 5 and the 3 strand are able to regulate gene expression and protein. So once this, we have the mature microRNA, this will be loaded into a protein complex called RISC complex. And the main protein here is argonautin-2. And this RISC complex, together with the microRNA, will be driven to the target message RNA. And will bind to the message RNA, inhibiting protein translation. And this will regulate the protein abundance. Or depending on how this matching happens, this binding happens, you can also induce mRNA cleavage, also decreasing the mRNA abundance and protein as well. So the hypothesis of our study was, well, since exercise induces so many changes in adipose tissue and cells have this mechanism of gene and protein regulation, we believe that exercise training would affect Dicer levels and microRNA biogenesis in adipose tissue. And this may serve as part of the regulatory feedback that could affect muscle function and exercise performance. So to start asking this question, the first thing we've done to understand if exercise could increase Dicer and adipocytes was to take black 6 mice, 8 weeks old, and put them to perform exercise on treadmill for 8 weeks. We also performed a maximum exercise test to prescribe the intensity, and they ran 6% of the maximum exercise test. 1 hour per day, 5 days per week. And after 24 hours from the last exercise bout, we sacrificed the mice to avoid any possible acute effect. And then we analyzed Dicer in different adipose tissue depots. So here you can see in epididymal fat, the Dicer abundance after aerobic exercise training, 8 weeks of exercise, it's 3 times increased. The same happens for subcutaneous adipose tissue. Here we have almost 2 times increased, and also brown fat, where we see the biggest effect. So after that, we were quite happy. Exercise can increase Dicer. we started to ask also if this is happening in humans. So we then took samples from men around 36 years to 63 years. This time they performed high intensity interval training where they were cycling for 1 minute and resting for 30 seconds. And this was performed 5 times. And the training was performed 3 times per week for 6 weeks. And then we measure, take fat biopsies and measure dicer expression before and after this exercise training. And here you can see in black, this is the dicer levels before 6 weeks of training. And we see that after 6 weeks, in the younger individuals, we see a drastic increase in dicer in adipose tissue. The same happens with older individuals in a smaller magnitude, but also happens. And this is interesting because we know that dicer is downregulated with aging. So, and this is associated with many metabolic defects. And then it's interesting to show here that exercise can increase dicer as well in older individuals. So maybe delaying the effects of aging. So our next question was the mechanism that could regulate dicer. So what happens when you do exercise in your adipocyte is basically exercise induced lipolysis, which will induce the breakdown of triglycerides and the release of free fat acids. This mechanism doesn't needs energy, but then part of this free fatty acids, as I mentioned before, is reuptaken and there is a process of esterification. So you can have more triglycerides just to keep inside the adipocytes or also to feed lipolysis. And this process expand energy. So it activates AMP kinase, which is an energy sensor for the cell. So our next question was if AMP kinase can regulate dicer in adipocytes. So we generated AMP kinase knockdown cell line, and then we treated these cells with adrenergic agonist isoproterenol and checked AMP kinase phosphorylation and dicer abundance over time. So here is what you see on the x-axis is time after time isoproterenol treatment, and then on the y-axis is the protein abundance. So this blue line here, it's the phosphorylation of AMP kinase, which goes up after an hour and then keeps high after 2 hours, and then after that starts to go back to basal levels. And interesting enough, you can see that dicer starts to increase after 2 hours, reach a peak after 4 hours, and then on 24 hours, it's almost back to basal levels. However, if we remove AMP kinase from the cells, of course, in this other blue line here, the dotted blue line, you don't see AMP kinase because AMP kinase is not there. And also you don't see dicer being increased. And even before isoproterenol treatment, just by not having AMP kinase in the cell, you also see downregulation of dicer. We next tried to test this model in vivo. So we generated a fat-specific AMP kinase knockout mice. We'll put them to do the same protocol of exercise, 8 weeks of aerobic exercise. And then we measure dicer expression in adipose tissue. And we can see that exercise can increase significantly dicer expression. However, when we go and check protein abundance, we see that in wild type, exercise increases dicer again more than 2 times. However, if you don't have AMP kinase in adipocytes, this increase is blunted. And this was quite interesting because here we can show that AMP kinase does not change this transcription of dicer, but AMP kinase is basically regulating possibly the protein translation. And there are some papers showing that AMP kinase can phosphorylate a protein, mRNA binding protein that is bound to the dicer mRNA. So when AMP kinase phosphorylates this protein, AUF1, this protein leaves dicer mRNA and then favors dicer translation. So we believe that that's the mechanism that's the mechanism happening in here. So far I showed you that exercise can increase dicer and it's via AMP kinase activation. So our next question was whether this mechanism was just an autonomous regulation of the cell, or if there was any endocrine factor that could also regulate dicer in adipocytes. So we have evidences in the literature that exercise induces a crosstalk between muscle and adipocytes. So we have the myokines, for instance, arisine, methionine-like protein, and kynureninic acid, which is known to be released from muscle and has a role in regulating adipocytes metabolism. And the opposite is also true. So there are adipokines and signaling molecules that are secreted from fat. They can also play a role in muscle metabolism. So our next question was to understand if there is any endocrine regulation of dicer in adipose tissue, and if this regulation is also dependent on AMP kinase in muscle. So to answer this question, we took advantage of a mice that is muscle-specific AMP kinase dead. So these mice overexpress AMP kinase, but AMP kinase doesn't work. And then we asked if by not having AMP kinase working in muscle, what would happen with dicer in adipocytes? So this work was done by Soren and then at the University of Copenhagen. So we had now 4 groups, wild type and AMP kinase dead, sedentary and exercise. And this time the exercise, we combined the voluntary plus the forced exercise in the treadmill. So these mice trained for 6 weeks. The treadmill was 1 hour per day, 5 days per week, and they had free access to the running wheel. And then we analyzed, after the end of the protocol, we segmented the mice and analyzed dicer abundance in subcutaneous fat. And here you can see that in the mice that trained, dicer is increased almost twice again. However, if you don't have AMP kinase working properly in muscle, you don't increase dicer in adipocytes. So after these results, we started to think that there might be a circulating factor that is coming out of muscle, and it's dependent on AMP kinase activation in muscle, going to the circulation and regulating dicer in adipocytes. So our next experiment was to exercise mice. We took the serum from this, from both sedentary and exercise. This was done by Soren and Thomas, again at the University of Copenhagen, and we checked for dicer expression. And at this time, we just did 3 days of exercise because we already knew that 3 days of exercise can also increase dicer. So as you can see here, if you take the serum from the exercise mice and treat cells for 4 hours, you already see an increase of almost 2 times in dicer abundance. We tried to test this in vivo as well. So we took the serum from sedentary and exercise mice and we treated sedentary mice for 3 days. And you check dicer abundance in adipocytes. And as you can see here, the mice that received the serum from exercise mice upregulated dicer more than 2 times. So from this part, we can say that aerobic exercise training induces muscle adipose tissue crosstalk to regulate adipose tissue dicer. And this mechanism is dependent on of AMP kinase signaling in both tissues. So since we saw all this regulation of dicer upon exercise, we started to ask ourselves whether dicer would be important for exercise. So our next strategy was to work with fat-specific dicer knockout mice. Again, the same exercise protocol, 8 weeks of exercise, aerobic exercise training. And we evaluated, we used this maximum exercise test to evaluate exercise performance. Basically, you put the mice in a treadmill, and each 3 minutes we increase the speed to the mice get to exhaustion. So we did this test before the 8 weeks of training and after that. So here it's in white is pre-training and in gray it's post-training. So you can see that the wild type mice increases from 16 meters per minute to almost 22. However, if you don't have dicer in adipocytes, you cannot improve your exercise capacity. And this was quite interesting because we are changing something in the adipocytes and we are seeing already this changing the performance. So our next question was whether this could be a problem in substrate metabolization. So we basically took 8 weeks old mice, and rather than training them, we just ran 1 exercise bout, and mice ran to exhaustion. We collected tissue and then performed an ex vivo substrate oxidation. So here you see valine oxidation in the skeletal muscle gastric nemmus. And you can see that right after exercise, valine oxidation is increased, but when we don't have dicer in adipocytes, you blunt the valine oxidation in muscle, which is quite interesting. You change something in fat, you see modifications happening in muscle as well. We also see that the glucose oxidation in the adipocytes is increased after exercise, but if you don't have dicer, this effect is blunted. So our next question was whether this decrease in glucose oxidation was because maybe the tissue is uptaking less glucose. So we performed a in vivo glucose uptake, and in here, we trained the mice for 8 weeks, and we performed this experiment. And surprisingly, we can see that either in epididymal fat and subcutaneous fat, knockout mice, they uptake more glucose. So we was wondering what was the destination of this glucose since it's not being oxidized. So then we checked the lactate levels in the adipose tissue fat pad, in subcutaneous fat. And we saw that when you take dicer knockout mice and put them to exercise, they accumulate significantly more lactate in adipose tissue. And that's interesting because we don't see any difference in the circulating lactate. It's basically what's going on in the fat. So our next question was to understand, well, dicer is important. If you don't have dicer, you don't run more. You change your metabolism toward to more glycolytic. So is there any microRNA playing a role in this mechanism? So we decided to perform a microRNA sequencing. And to do this, we again went back to the AMP kinase, muscle-specific AMP kinase dead mice, because they do express dicer in adipocytes, but they are not sensible to increase dicer upon exercise. And we performed these microRNAs in a subcutaneous fat coming from wild type and AMP kinase dead mice. So here you see this heat map where red is upregulated, green is downregulated. Each row is a mouse and each column is a microRNA. And here we selected only the top 50 upregulated microRNAs compared to wild type. So you can see here that exercise really increased significantly the expression of microRNA in adipocytes. And this is not very clear when you don't have AMP kinase, actually. You don't see this regulation happening. So just having this Venn diagram, which I believe show it better. So you can see here that approximately, well, 50 microRNAs are increased in the wild type combined aerobic exercise training, while you don't see any, just one in the AMP kinase that trained, and 3 in the AMP kinase that's sedentary. So it's really, exercise is really regulating dicer, and this is really affecting the processing of and expression of microRNAs. So among of this 50, we went over and characterized the microRNA 203 because we started to see that even if any kind of intervention we've done, either caloric restriction or obesity, whenever you have upregulation or downregulation of dicer, you see this microRNA being also regulated in the same direction as dicer. So we took this microRNA and overexpressed it into TT3L1 differentiated adipocytes and treated these cells with either vehicle or isoproterenol. And you see when you treat adipocyte with isoproterenol, you see an increase in lactate concentration in the media. However, if you overexpress this microRNA, you kind of blunt in half of this increase that happened in the scramble cells, indicating that this microRNA is regulating glycolysis. We checked a couple of glycolytic enzymes, and the most that were more striking is the PGAN, which stands for phosphoglycerate mutase. It's one of the last enzymes in the glycolytic pathway. And you can see here, if you treat cells with isoproterenol, you don't see much change in the expression of this protein. However, if you overexpress this microRNA, this protein is almost gone. According to microRNA database that can predict targets, PGAN is not a target for miR203. And so far, we still don't understand, we still don't know if miR203 is binding to PGAN or other PGAN regulator, and that's why we see this effect. But indeed, we see that miR203 is regulating glycolysis. So if you delete dicer in adipose tissue and do exercise, you see impaired exercise performance. You see decreased skeletal muscle and adipose tissue oxidative capacity, increased adipose tissue glycolysis, and also we observed that aerobic exercise induces the expression of dicer and miR203 to control glycolysis in adipose tissue. So to summarize, or actually to conclude, when you do exercise, you're going to activate AMPK in muscle. So in this will trigger glycolysis, oxidation, and activation of TCA cycle to increase ATP levels. Also, the tissue will secrete a molecule that so far we still don't know which one it is, that goes to fat and upregulate dicer. At the same time, the adrenergic signaling will activate AMPK in fat, which will increase dicer expression to favor oxidative metabolism, and also to increase miR203 to blunt glycolysis. So AMPK is a protein that is energy sensor from the cell, and dicer is a hub for the cell where you can regulate the balance between oxidative metabolism and glycolysis. And if you remove this from fat, you're not going to have fat that is able to give the right amount of substrate and also a healthy fat that is important for exercise performance. So with that, I finish my presentation and I'm open for questions. Thank you very much.
Ariana, do you want to introduce Mart?
Oh, hi. Yeah. Got it. So now I'll be presenting Dr. Moji Rado from the lab at the Institute of Cardiovascular Intervention at Ludwig-Maximilian University in Munich, who will be presenting her work on novel transcription and insight into adipogenesis for heart development.
All right, thank you very much. But don't we give the chance to Branda to answer to the questions? Or, okay.
No, we'll have like 15, 20 minutes open for questions at the end.
All right, then I share my screen.
Great.
Then good afternoon, everybody. Well, at least here in Munich, it's afternoon. I don't know what is it at your place. So I would like to thank the organizer to invite me and to give me the chance to present my work today. So I'm very glad. I'm Moji Rado, so I'm a postdoc in Munich in Alexander Bartelt Lab at the LMU. And the work that I'm going to introduce you today is actually a joint project between Herzig Lab at the Helmholtz Center in Munich and Alexander Bartelt Lab. And I'm going to talk about novel transcriptional insight into adipogenesis from heart development. So I have a first slide, so summarizing my CV, because I've been asked for that actually. And well, so I'm French. I started actually my master at the University of Birmingham in England when I was working on cellular entry of nanoparticles. I defended my master thesis at the University of Montpellier, which is in the south of France, about a work on alternative splicing of the oncogene Yorkie. And then in 2012, I actually started my PhD thesis at the University of Nice, very nice city also in the south of France, where I worked also on microRNA and adipocyte, but plasticity this time, so on browning basically. And since 2016, I'm here in Munich doing my postdoc, where I'm working on molecular regulatory circuits in lipid homeostasis and adipose tissue biology. So the work that I'm going to show you today is actually available now on bioRxiv, but we are still working on it. So actually, I'm going to start this presentation by a question that actually you all have the answer. What is obesity? So we actually all know that obesity is an expansion of the adipose tissue. So either by accumulation of the lipids in the already present adipocytes, or by recruitment of new adipocytes, either both. But what is actually very interesting to know is that what adipose tissue is, because you know that we have several types of adipose tissue. So there are the brown adipose tissue, which are which are razor scars in humans. So they are localized in the cervical, supraclavicular, and paravertebral area. And so there is also the white adipose depot, which are subcutaneous. So we are going to talk mainly today about the subcutaneous, abdominal, and gluteal. Oh, maybe I can, yeah, maybe you can see here. So abdominal and gluteal, or intra-abdominal or visceral white adipose tissue, which can be omental, retroperitoneal, or visceral. So then there are different types of adipose tissue. So what's the difference between those adipose tissue, in addition of being like anatomically differently located in the body? What could be the difference in the origin actually of those adipose tissue? Oh, wait a minute. Here we go. Yeah, great. So actually another question so is like, from a developmental point of view, are they different? So when we look at the 3 germ layers at the very beginning of the development in general, all the adipose tissue are coming from the mesoderm, as well as heart or muscles or bones, for example. And this germ layer, so the mesoderm, is going to differentiate in several layers, so like the somit, the intermediate, or the lateral plate. And the somit are actually going to give the somatic progenitors, which are where the brown adipose tissue is originate from, and the lateral plate going to give the splanchnic progenitor, where the visceral white adipose tissue is coming from, or the somatic progenitor, which is more the progenitor for the subcutaneous white adipose tissue, among other tissue, of course. So there is definitively something different between all those tissues. And we can also see that in case of obesity, the accumulation of either subcutaneous white adipose tissue or visceral white adipose tissue is going to give a different metabolic answers. So we all know that like metabolic healthy patients are actually accumulating fat more in the subcutaneous white adipose tissue, more in the lower body, so in the gluteal subcutaneous adipose tissue, giving this rather pear shape. When metabolically unhealthy patients, which are around like 80% of the patients, are going to accumulate fat more in the upper body, in the visceral white adipose tissue. So of course, many labs got interested, like why there is so much difference in between the accumulation of those 2 types of fats. And the laboratory of Matthias Bluer, actually in Leipzig here in Germany, investigate the difference of methylation between the subcutaneous adipose tissue and the visceral adipose tissue in obese and non-obese patients. So they compare actually 2 different cohorts, and they could show that HEN2 was in the top 3 differentiated, differentially expressed gene between the subcutaneous adipose tissue and the visceral. In parallel, we're actually looking at the difference of gene expression between subcutaneous white adipose tissue and brown adipose tissue in humans. So we found that HEN2 was almost not expressed in the brown adipose tissue, and in collaboration with Matthias Bluer, we could confirm his data showing that indeed HEN2 was more expressed in the visceral. Those data are also confirmed in mouse model, which was very interesting. And in addition, we could see that HEN2 was inversely correlated with BMI in human and with body weight in mice, so making its expression actually correlated with the obesity status of those patients. So what is HEN2 actually? Because HEN2 in adipose tissue, there is almost no publication about it. So HEN2 stands for heart and neural crest derivative express transcriptional factor 2. So it's nuclear, it's usually dimeric. It's member of the twist family of basic helix loop helix, so BH-LH proteins, and it binds most of the time the E-boxes. So having that said, so that doesn't say that much about HEN2, but there, if you just check HEN2 in PubMed, there are many publications in HEN2 in cardiac development and neurogenesis. And it has been actually reported that HEN2 might be regulated by the BMP4, so which is the morphogenic protein 4. So now we're wondering, since HEN2 we see HEN2 regulated in the adipose tissue, so what about the relationship between HEN2 and mesenchymal precursor? And actually, there are few publications, so 2 publications, one relating HEN2 with osteogenesis, so HEN2 would inhibit RNAs2, and then impair osteogenesis. And in a model of cardiomyoblasts differentiated from adipose-derived mesenchymal precursor, so HEN2 was used as a positive marker. And when we looked in animals, we could see actually that HEN2 was, of course, highly expressed in the heart, but then after that, in different tissue, it was quite highly expressed in the gonadal, as I show you, but yes, not expressed in muscle or in brown adipose tissue. So that was rather surprising because we know that brown adipose tissue and heart share more developmental, how can I say, common point than the white adipose tissue. So let's go through that again. So just to remember, so the mesodem part during development going to give multipotent mesenchymal precursors. And those precursors, they are able to differentiate either in white preadipocyte, in myogenic progenitor, chondroblast, or osteoblast progenitor. The white preadipocyte most likely are going to give white adipocytes. And the myogenic progenitor, actually, they are progenitor for the brown adipocytes. Or for the muscles, so different type of muscle. There is also the smooth muscle, which are not here. And so those are going to give chondroblast and osteoblast. So once again, here we can ask ourselves, then what brings this common point between white adipocytes and cardiac muscle? And actually, when we look at the compound which are inducing this differentiation, we could see that BMP4, sorry, was commonly a common factor inducing the differentiation of those 2 cells, while the brown adipocyte would be potentially more induced by BMP7 and skeletal muscle, like other compounds. So that'd be the reason why we see this correlation with white adipocyte and cardiac muscle. But today we are going to focus more on this part, which is actually adipogenesis. So adipogenesis, as we see, a multipotent mesenchymal precursor. They can give several type of cells, including adipocytes. And all this process from multipotent mesenchymal precursor to mature adipocytes is rather separated in several steps. So the first step is the commitment into the precursor to a pre-adipocyte. Then the pre-adipocyte is going to stop growing and then being induced in what we call differentiated adipocytes, and the differentiated adipocyte going to maturate to become a mature adipocyte. Then the question is, let's go back to our gene of interest, HEN2, how HEN2 is actually regulated over this adipogenesis process. And so BMP4 is inducing commitment. Oh, sorry, I've forgotten something. So the differentiating adipocytes are characterized by some differentiation marker, which are PPAR gamma, CBP alpha, CBP beta, among others. And the mature adipocytes are characterized by the expression of adiponectin, leptin, and perilipid. So then, another question, how HEN2 is regulated over this full process of adipogenesis. And actually, we use HMAT cells, which are a model of human multipotent adipose-derived stem cells. So they are already pre-adipocytes. And so using like a normal cocktail of differentiation, we could see that HEN2 was really increased at day 3 of differentiation and then was decreased later on. So knowing the developmental role of HEN2, we decided to actually inhibit HEN2 at day -2, so before the induction of the differentiation, and look at how adipogenesis was affected by this loss of function, this early loss of function of HEN2. And actually, we could see that the loss of function of HEN2 was completely impairing the accumulation of lipids. So this already staining show you that clearly we don't see any lipids when HEN2 is inhibited. looking at early differentiation markers, so as PPAR gamma, CBP alpha, or PPAR GC1B, we could see that all those genes were significantly decreased under the loss of function of HEN2. And when we looked at transcriptional analysis, we could see also that many genes implicated in adipogenesis were decreased, including perilipin and adiponectin, which were the most regulated gene in that case. So that was very interesting. Nevertheless, we were asking us this other question. So you can see at day 3, so that is the same graph than earlier, there is this increase of HEN2. So where does that come from? And actually, that between day 0 and day 3, we had this cocktail of differentiation, which is made of several compounds, including so dexamethasone, rosiglitazone, insulin, transferrin T3, and IBNX. And we could see that actually only dexamethasone was able to induce the expression of HEN2 as much as the normal differentiation media. So what is dexamethasone? Dexamethasone is an agonist of the glucocorticoid receptor. So the next step was then to inhibit the glucocorticoid receptor and see if we could keep seeing this increase of HEN2. And actually by transfecting siRNA against NR3C1, which is the gene coding for the glucocorticoid receptor, we could see that we were completely impairing this HEN2 response under DEX treatment. So those findings just confirm our hypothesis that there is this glucocorticoid HEN2 pathway. But then what happened in vivo? So that was definitely our next questions. And actually an acute injection of dexamethasone in animals so in mouse, was also showing an increase of HEN2 expression in the gonadal white adipose tissue. And when we looked at differentiated adipocytes from the stromal fractions, we could see that in mice which were knockout for GR and treated with DEX, we were completely impairing this increase of HEN2 under DEX treatment. So that was confirming also our hypothesis that it was these GR-HEN2 pathways. But then what this GR-HEN2 pathway might regulate? So that was our next question. So for that, we realized RNA-seq so in Hashmat cells, and we treated those cells with dexamethasone and transfected either with SI control, SI-NR3C1, or SI-HEN2. And actually, we looked at the gene which were commonly regulated by the inhibition of NR3C1 or HEN2. And you could see with this principal component analysis that the gene knockdown for GR were nicely clustering with the gene not treated with dexamethasone. So that just confirmed the validity of our model and how the cells were very well answering to DEX and to the SI. When inhibiting HEN2, so the effect were rather smaller, but we're not expecting anyway HEN2 to be as efficient as GR. Having that said, we just checked also for pathway analysis. So the pathway analysis at the intersection, as I mentioned, and we could see that pathway as mTOR signaling or GPCR were affected, and those pathways have been previously actually described as playing a role in adipogenesis. So that was confirming like our hypothesis. So now what the outlook for this project? So basically in adipocytes, we showed that it was a GR-HEN2 pathway. So we did this nice pathway analysis, but then what are the direct targets for HEN2? So we are currently doing chip sequencing on knocking animals, so HEN2 triple-fag, which would allow us to define direct target of HEN2 in adipocytes. And then in heart, so what could be the outlook there? So is there a GR-HEN2 pathway in heart? So that we don't know. There are no literature actually relating GR to HEN2, but we have like very preliminary data showing us that dexamethasone also induce the expression of HEN2 in primary cardiomyocytes. So it could be something interesting to check there. So then if there are like 3 things that you should remember from my talk today. So the first one is that the glucocorticoid receptor regulate HEN2 expressions, and that HEN2 affects adipogenesis. And the last thing, but not the last, it's that this work would not have been possible without the help of many of my colleagues from Herzig Lab, and the help from Sajjad Kani and Stefan Kochi from Alexander Bartlett Lab, and also many collaborators, including Matthias Bluer, who just provided the human samples, Kirsi Aviatanen, who provided the brown adipose tissue from humans, and many others who helped me to achieve this work. And I would also like to thank the Alexander Van Humboldt Foundation, who was like kind enough to give me a grant for a fellowship for 2 years and who paid my work, and myself. Then thank you very much for your attention. I'm open for questions, if we have time.
Yeah, we've got some time. Thank you, Madame Bruna, for 2 great presentations. So I'll just have a look through the Q&A, and we'll give it a few minutes for some to come in for Maud. I've got some, and we'll start off asking Bruna some of the questions that are in. So someone was asking what about the effect of exercised serum from AMPK knockout adipocytes and the response to DICER levels? Did you look at that?
Yeah, yeah, that's a quite interesting question. Thank you for that. And actually, we did not look for that, and this question makes total sense, because our idea is that there is a signal from that is dependent on kinase in muscle that is inducing DICER in fat. But we don't know, we know that we need kinase in fat, we know that we need kinase in muscle, but we never took the serum and treated cells that don't have kinase, for instance, the adipocytes that don't have kinase with the serum of trained mice. So we don't know if there is any molecule there in the serum that actually is activating kinase, therefore inducing DICER. Yeah, this it's in the to-do list for next study, but that's an interesting question.
Great. So what else do we have in terms of? So yeah, so is DICER related? Fatima Perez de Heredia was asking if DICER is related in any way to hypoxic pathways or to MCTs.
Well, we don't know so far. So the last part of my presentation where I showed PGAN being downregulated upon microRNA 203 overexpression, we believe that HIF1 alpha seems to regulate PGAN. So there is something there. We believe that there are this possibility, but it never tested. So yeah, that's the short answer for that.
Great, thank you. So we had a question from Craig Daly. Interesting talk, thanks. You showed beta adrenergic stimulation increases DICER, and it's known that alpha-2 adrenergic stimulation decreases lipolysis. Do you know if A2 stimulation will also increase DICER at all?
Well, we never blocked lipolysis to see if there is any regulation of DICER. We try to activate kinase in other ways, for instance, with ACAR and also metformin. That in the case of ACAR goes straight to the kinase activation, and we still see, and we do see DICER upregulation. So I don't know this. I don't know if it would decrease DICER expression, but I believe that the increase in DICER expression is not only dependent on lipolysis activation, it's more dependent on M kinase activation. So whatever you activate M kinase, or you regulate DICER. If you block M kinase, you downregulate DICER. That I showed. I showed this with the M kinase knockdown cells. If you don't have, just by not having M kinase, you downregulate DICER. So, but we don't know about blocking lipolysis.
Great, thank you. Ariana, do you have any questions for Bruno?
Oh, okay, yeah. Sorry, I haven't I haven't really cleared this. Like, have you just checked all, like, the dicer? Have you just checked it in white adipose tissue? Did you check it in brown adipose tissue as well, like, throughout all your work?
Well, the only thing we check many things in the dicer knockout. We check the glycolytic genes, we check the lactate and the glucose uptake. Glucose uptake we've done in all 3 depots. Most of the gene expression and all the work was done using subcutaneous fat, because if you look in the dicer knockout mice, this mice is lipodystrophic. So you see a decrease in the epididymal fat and enlargement and whitening of the brown fat. So the depot that is less affected by the knockout is the subcutaneous fat. So that's why we focus mainly on that depot to just avoid any effect of the lipodystrophic phenotype. But yeah, it's mainly white subcutaneous fat. Thank you.
Thank you.
So I think, yeah, so there's another couple of questions. And I've actually got a couple as well. So there's one from Patricia Molina, who asked, did you look at the mitochondrial content following exercise in the AMPK dead muscle?
No, actually, the only thing we did with the AMP kinase that muscle-specific AMP kinase that mice was check fat parameters. So it's been published already, the characterization of these mice. Don't remember now if there is any change in mitochondrial biogenesis. I'm sure that they run the same, because this was also an issue. If they would run less, or this would be a problematic, right? Because maybe dicer is just not being upregulated because you're not running enough, but they have the same running capacity as a wild type mice. I'm just not aware if there is any change in the mitochondrial profile of this mice, and we never tested it. Thank you.
Great, thank you. So I just had a couple of others. I can't remember if you mentioned it, but what muscle was it? And is there a difference between like slow, fast twitch? And is the microRNA profile different from these types of muscle? Do you know?
Wow, yeah. Actually, I cannot reply for sure. What is skeletal muscle? It wasn't specific to glycolytic or oxidative. I believe was for both. We never checked the microRNA expression in these mice. From my study in the wild type mice, what we found is that dicer is not increased in muscle after exercise. And this is intriguing, because if AMP kinase is regulating it, why it's not regulating in muscle and only in fat? So this is something that so far we don't understand, why dicer is not increased in muscle after exercise. So yeah, we never checked the microRNA content in this kinase that muscle mice.
Okay, and then just I think the final one. So given the significant changes in exercise performance that you see, do you think these microRNAs could be affecting cardiac function at all?
Well, yeah, that's a possibility. We never focus in the heart of these mice. We know from previous studies from Dr. Khan's lab that fat is the main tissue that can secrete, for instance, microRNA. These microRNA 203, we did not publish this, but we have some data where we analyze circulating microRNAs, and these microRNAs not increased after exercise. So it seems that this specific one, it's working on the fat and not being regulated. But I would bet that there are many microRNAs coming out of fat after exercise and going to different tissues, and heart, of course, can be a possibility.
Okay, great. Thank you. Thank you for that.
Yeah, thanks for the question. Yeah.
That's all right. Ariana, do you want to ask the questions to Maud, and if you have any as well?
Sorry, the questions from the question and answer box.
Yeah, so.
Yeah, sorry.
No, that's right. Otherwise, it's just me talking all the time, and no one wants that.
Right, so the first question I see here is from Magnolia Rizagneta. That's wonderful presentation, thank you. And it's that she asked a question for Maud: if visual what adipose tissue in the unaltered type, what do you think is the next step in target intervention with all the knowledge recently generated?
Yeah, so yeah, thanks for this question. It's a rather broad question, and what I wish I would have the answer. And I guess we have like another question which is in that type earlier. Yeah. So how we can control basically the fat deposition. I would not have an answer for that today. So that's why actually so many labs are just trying to understand what is the genetic difference between those two tissues and why different individual has this capacity to store the fat more in the subcutaneous or in the visceral. So there is also a gender bias here. So female would have also the tendency to store more the fat in the subcutaneous adipose tissue compared to the visceral. So regarding the second part of the question, so what would be the next target and what would be the next intervention? So we are not there yet. So I mean, right now we know that the most efficient way to kind of cure obesity, it's a bariatric surgery, which is rather very invasive and yeah, not optimal. So that's why we are keeping looking in that direction. But I would not have like a very specific answer for this question, which is actually very interesting, and that's why we are keep looking for in that direction.
Okay. So moving on to the next question from Takesh Patel. She asked, have you checked HAN2 expression in fraction of what? Is it expressed in mature adipocytes or pre-adipocytes?
Yeah, so that is a very relevant question from Satish. So actually we did look at it, and HAN2 is indeed expressed in the adipose fraction. Even so, it's slightly more expressed in the stroma vascular fraction. Which confirms also our hypothesis that HAN2 has a rather early role in the development. But yes, we do see HAN2 in adipose tissue in the adipose fraction.
Great, thank you. Another question. So many adipokines from fat cells has made me wonder if there could be subtypes of white adipose tissue and brown adipose tissue. Do you have any thoughts about that?
Well, I'm not sure I understand the question. You just removed it already, so.
No, I think it's still there.
Yeah, you're right. Yeah. So many adipokines from. I mean, so if it could be subtypes of white adipose tissue or brown adipose tissue. So I guess that's one of my first slides answer to this question. So subtypes of white adipose tissue, yes, definitively. We saw that there are different type of white adipose tissue, subcutaneous, visceral, and plus other one. And brown adipose tissue. So for now, brown adipose tissue, it's I would say, yeah, there is the brown adipose tissue, but also what we call the thermogenic adipocytes, which are more like adipocytes, which are able to, I did not develop that point, but brown adipocytes are able to release energy instead of storing it. And then they would come from white adipose tissue. So it's what we call like beige adipocyte or bright adipocyte. So there are definitively subtypes of those tissues in the body. Yeah.
Okay, so one last question from Anita. She asked, referring to glucocorticoid effects on AMP2, is there a variation in expression based on age?
So on glucocorticoid, yeah, so there is definitively variation of expression of the glucocorticoid. Referring to aging, I'm not 100% sure, but referring to the obesity status of the patient, yes. And if we think also about development, there are also difference of glucocorticoid expression. So there, what can I say? During life, there are differences. With aging, I'm not so sure. I would say potentially, but I could not answer 100% that yes, with aging, glucocorticoid increase or decrease. I'm sorry for that.
Okay, I also have a question myself about your in vivo part of your study. Did you use animals from both sexes, or did you just use male animals? Or as we know, like sexual differences comes like in terms of hormones as well as cardiac health as well, like, because like, you know, in human population, females usually have less cardiac issues than males. Did you use one?
So actually, so that's a very interesting questions. I mean, there are many data that I did not show. I also have like adiponectin-free hen2 flocks animals where I don't see so much phenotypes, but indeed I see this increase of hen2 mainly in females, and it's not so it's not as significant in males. So they are definitive. I mean, in terms of expression, it's not so different between male and females, but in terms of answer to glucocorticoid, yes, it is. So I guess there is definitively like hormonal regulation there who could affect hen2 expression when animals are treated with DEX.
Great, thank you for that. Thank you both for your time answering questions and for 2 great talks. And thank you for Ariana for co-chairing today. It's been a great session. And I'm sorry if we've missed a couple of questions. The time's almost up, just up. But I hope you'll all join us next week for our muscle physiology and obesity session. So thank you very much, everyone. Take care.
Thank you a lot.
Thank you.
Thank you. Bye.
Thank you. Bye-bye.